Hello, and welcome to Spotlight on Science from St George's University of London. My name is Julian Ma, and today in this third video on coronavirus, we're going to be thinking about the progress that's been made with the vaccines against COVID-19. Today is the 28th of May, and in the UK we are well into the 10th week of lockdown. And our thoughts have turned to the progress that we're making with the vaccines and how soon we might have one. Over the last few weeks, we've heard about over 100 candidate vaccines which have been developed by various groups around the world. And in the UK, there are two lead candidate vaccines, one developed from the University of Oxford and one from Imperial College. Later on in this video, you'll be hearing from Professor Paul Heath, who runs the Vaccine Institute at St George's, which is one of the clinical trial sites for both vaccines. We'll also hear from Fran Mabesa, a research nurse, and she'll be telling us what it's like to be a volunteer on the vaccine trials. And then we have the two chief scientists from those two vaccine development teams, Professor Andrew Pollard from the University of Oxford and Professor Shattock from Imperial College. But before we start, I would like to just get us all onto the same page as to what a vaccine is and what we're trying to do with it. Essentially, vaccines are mimics for pathogens, and they stimulate our immune systems to provide long-lasting protection against diseases. We're all familiar with vaccines. We've had them as children. Some of us have had them as adults if we've travelled abroad. And at the moment, there are about 26 vaccines against major diseases. And those include well-known diseases, which we've almost forgotten about in the West: polio, tetanus, whooping cough, diphtheria, and so on, because these vaccines have been so successful in reducing the incidence of those diseases. But there are still important diseases that we don't have vaccines for. HIV stands out, of course. Tuberculosis protection in adults is very difficult. And of course, the one that we all want at the moment is the one against COVID-19. So how do vaccines work? Well, vaccines activate the immune system, which in essence is the white blood cells in our bodies. So we have white blood cells which are scattered around in our blood, and you can see in this picture here all the red blood cells and these two white blood cells. Those are lymphocytes, and those are the cells that we want to stimulate with our vaccines. There are also white blood cells distributed throughout our bodies, but they're really highly focused in the what's called the lymphatic system, namely the lymph nodes, the spleen, and tonsils. And that's why, for example, when you get an infection or you get a sore throat, for example, you get raised lymph nodes in your neck because the white blood cells in those nodes have been activated. Once the white blood cells have been activated, then they really work in two main ways. There is a group of white blood cells which will go on to produce antibodies, which will neutralize or kill virus or bacteria. And there are other white blood cells called T cells, which can go ahead and kill infected cells directly. Now, the whole thing about vaccines is that they allow us to react stronger and faster when we encounter diseases if we've been vaccinated against them before. And this is shown in this rather complicated slide here. To make things simple, on the y-axis, just think of this as your immune response, and on the x-axis, think of that as the time. So when we first are vaccinated, our immune systems respond to the vaccine, and it generally takes about 10 days for the response to ramp up and get to its maximum response. And then we are protected against the disease. After about 10 days, our response wanes, and it actually wanes quite quickly, and we get down to a basal level. But the important thing is that we now have memory. We have white blood cells which are going to remember that specific infection, so that if we ever encounter it again, and this time this is the proper infection, then our white blood cells are activated much faster, and you can see how much steeper this curve is here, and we get a much larger response much faster. And that's what protects us against the disease. And then you'll see as we recover from the infectious disease, our immune response again wanes, but it comes down at a slower rate, and it stays at a higher plateau level than before, ready for the next time. So that's what we're trying to do when we vaccinate against diseases. We're trying to make sure that we can respond quickly and we're trying to make sure that we have long-lasting immunity. Now, what are we looking for in a good vaccine? Well, in general, vaccines have to be acceptable, affordable, and accessible. And I think it's true to say that no vaccine is perfect, but actually many of the ones that we have are pretty close. So what do I mean by acceptable? Well, a vaccine has to be safe, of course, and it has to have ideally no side effects, or if there are side effects, they have to be acceptable. It needs to protect a high proportion of patients against the disease, and it needs to work in all of our target populations. We've seen in COVID-19 how the elderly are particularly prone to serious consequences of COVID-19. So the vaccine has to work in the elderly as well as in younger adults and in children. It needs to be easy to administer. We don't want a vaccine that requires multiple doses before it starts to work. And once you're vaccinated, ideally you're going to have long-lasting protection. And with a disease like COVID-19, we also have to think about how we're going to deliver the vaccine to the rest of the world. And therefore it has to be affordable and accessible. It needs to be low cost to produce. We have to think about people in less developed regions of the world, low and middle income countries, and how we're going to make an affordable vaccine for them. We need to be able to manufacture at very high volume. There's a population of around 8 billion people in the world, and most of them are going to want a COVID-19 vaccine. And thinking about the different regions in the world, a vaccine has to be easy to store and transport to different areas of the world. Well, that's probably enough from me for the time being. Let's go and meet some of the people who are actually working on the COVID-19 vaccines. So to begin with, we're going to meet Professor Paul Heath. Paul is the Professor of Paediatric Infectious Diseases at St George's, and he's also the Director of our Vaccine Institute. Paul, thanks ever so much for joining us today.
Thanks, Julian.
You're in charge of our Vaccine Institute, which is doing all the clinical trials, the vaccine trials for coronavirus. Tell us a bit about the Vaccine Institute and what it does.
The Vaccine Institute is purpose-made for clinical trials. Those trials have been in adults, prominently, but also we do many pediatric vaccine studies. Often they're done in the family home, but sometimes we also do them in the Vaccine Institute. And the other group that perhaps more recently we've been focused on is pregnant women.
Okay. And can you tell me something about how you recruit volunteers into the trials?
Yeah, sure. A whole range of options are available. And so, for example, for our current trial, the COVID vaccine trial, that is predominantly trying to recruit healthcare workers. So we're recruiting through the hospital and the university through specific emails that go out to all of these groups. But it's also through our media officer. It is also directed to the local media, local press, newspapers, radio stations, and then it spreads far and wide thereafter.
So for the current coronavirus trials, anybody who fits your criteria can volunteer. And how do they do that?
For volunteers to recruit to this COVID vaccine trial, Julian, they would go to the Oxford University COVID vaccine trial website. Then they have a number of options for where they would like to recruit through. There are 4 trusts in the London area, of which St George's is one. So we obviously would like St George's hospital and university staff and local people to be recruited through our site.
Sure.
Yeah.
I see. So I understand that the Oxford trials have been going on apace, and that you're now entering what's called phase 3 trials, and you're shortly going to be starting a different vaccine from Imperial College, which is a phase 1 trial. Can you just explain what the difference between a phase 3 and a phase 1 trial is, please?
Yeah, certainly. The phase 3 trial that we're doing with the Oxford vaccine is also called an efficacy trial. So it really is trying to see whether the vaccine is effective at preventing the disease. So it's a blinded, randomized control trial. So half of the vaccinees are receiving the COVID vaccine, and the other half are receiving a control vaccine, which in this case is a meningococcal quadrivalent conjugate vaccine, so a vaccine that is effective against 4 serogroups of Neisseria meningitidis, a vaccine that we know to be safe and we know to be effective. And of course, those that receive the meningococcal vaccine, we would not expect it to be protected against COVID. So the efficacy trial, if suitably large, and it does need to be large, will ultimately demonstrate whether this vaccine, this COVID vaccine, can prevent COVID in the recipients of that vaccine. Okay. And also being large, it provides a lot of confidence to us of the safety of the vaccine, because it will identify whether there are any important safety signals. Okay. So that's the phase 3 trial. The phase 1 trial, of course, is the first in human trial with such a vaccine. And that's done in a much smaller number of recipients, perhaps 20 to 50 of that order. They will be healthy adults. And the purpose of that is to look at the safety of the vaccine when used for the first time in humans.
So the Oxford trial has been through that earlier on?
Absolutely, yes. You say the imperial COVID vaccine is now moving into that stage, and that will be a small number of subjects, then moving on, we hope, if there are no concerns with regard to safety, to the phase 2 trial, a much larger number. And then if that shows some suggestion of immunogenicity as well as safety, then it would move on to the phase 3 trial, the trial that we're now doing with the Oxford vaccine.
I understand. Thank you. And just finally, I'm kind of interested in how much it takes to run these clinical trials. I mean, you know, the phase 1, which is the smaller one, roughly how much does that cost?
Oh, how much does it cost to run a phase 1 trial? I can't really give you a cost for a phase 1 trial. I don't think.
Perhaps a ballpark? Are we talking like tens of thousands, 100,000s?
We're talking hundreds of thousands, certainly, yes. And then, depending on the number recruited, of course, it will be millions for the phase 3.
Yeah.
So the whole program is, of course, extremely expensive, but
worthwhile. Yes, absolutely. That's great. Thanks ever so much for talking to me today, Paul. And I'd like to wish you all the best for the current trials and those which are coming up soon. Thanks.
Thank you very much, Julian.
Well, next I'd like to welcome Professor Andy Pollard. Andy is the Professor of Paediatric Infection and Immunity at the University of Oxford. Andy, thanks very much for finding the time to join us.
No problem. Good morning.
Andy, one of the impressive things about the vaccine that you're testing at the moment is the speed at which you've managed to get it into the clinic. How did you manage to do that so quickly?
Well, I think there's several reasons why we've managed to go so quickly. And the first actually is because of the huge knowledge that there is already about the biology of coronaviruses. We've had 2 big epidemics over the last 20 years, the first in 2002 with SARS coronavirus 1, and then subsequently from 2012 with the MERS coronavirus. And although both of those outbreaks were contained using public health measures, a lot of effort went into vaccine development. And there were a large number of studies, mostly in animals, but some human studies, that provided a lot of data about what sort of protective immune responses might look like. So when this new coronavirus came along and the genetic code of the virus became available in January, it was possible with the team here, led by Sarah Gilbert, to essentially adapt the vaccine they were already testing for the MERS coronavirus and to add in the particular gene from the new coronavirus that's needed in order to make immune responses.
I see. So can you tell us about the vaccine and how do you expect it to work?
Well, what the vaccine is it's actually made from a common cold virus called an adenovirus, which has had part of its genetic code removed so that it can't make any new viruses. So when it's used as a vaccine, it's alive, but it doesn't then go on to cause an infection, which makes it completely safe. And what we've done with this virus is taken the genes which are used to make spike protein from coronavirus and put them into this common cold virus so that when that is used as a vaccine, we make immune responses against the critical part of the coronavirus.
I see. And That sounds like quite a high-tech vaccine compared to the conventional vaccines that we're more familiar with. Why have you gone that route straight away and not tried the more conventional approaches?
Well, actually, around the world at the moment, there are said to be 150 different vaccines that are in various stages of design and development. 10 of those already in clinical trials. The vast majority of them are actually using new types of platform technologies. And the reason for that is if you already have a technology which allows you to move very quickly to insert, for example, a new gene, you can get into clinical trials much earlier, which of course is necessary in the urgency of a pandemic. There are some of the vaccines in trials are using conventional approaches, killing the virus and using that as a way of making a vaccine, or even just taking proteins which the virus makes and using that as the way to make the vaccine. Those approaches are being tested at the moment, and it will be very interesting to see which of the very different technologies being used are going to provide the best level of protection against this virus. I think we need lots of technologies out there to find the winner.
Sure. And
or in fact, I should say the winners, because I think the world, the best thing is to have multiple different hits on the target.
Now, you mentioned that you're using the common cold adenovirus as a sort of backbone for the SARS-CoV-2 vaccine. What happens if you've already been infected with that adenovirus?
Well, there are actually several of these adenoviruses which are being tested or will be tested soon as some of the different developers. The particular one we're using here is actually an adenovirus which it causes colds in chimpanzees. So it's a chimpanzee adenovirus. And so human populations have very rarely seen this particular one. And so we don't already have immune responses to that virus.
Oh, I see. So in future, if you need a second round of immunizations for SARS-CoV-2, would you be able to use the same vaccine that you're giving the first time?
Well, I think if we went back a few years, conventionally people would have said you can't use the same adenovirus twice. And perhaps because a lot of the early work was done with human adenoviruses where we already had immunity. And when you made a vaccine out of one of these, you just boosted that immunity. So a third go was extremely difficult to do. But we have quite good data now, I think, with some of these other adenoviruses, which humans haven't seen before, to show that you do get boosting when you give second doses of them.
I know that the vaccine trials have been going on really fast. What results have you had so far?
Well, we're not in a position today to share any results. We actually, as you say, have trials that are currently in flight. And to maintain the integrity of the trials where the volunteers are all blinded, we're not releasing data. I mean, the trials are progressing well. We've recruited the phase 1/2 trial, which are the first-in-human trials, and then an expanded sample size of adults, over 1,000 people vaccinated. And we're now moving into the next phase of trials, which includes older adults, where we know it's much tougher to get good immune responses, so we're studying them in great detail. And then a much larger study to find out whether the vaccine actually protects people, and that's currently focused on hospital workers, primary care staff, and people in the front line of the fight against coronavirus.
Okay, so you're not studying all age groups yet?
No, we're starting with younger adults, and the definition of that is the under 55s. And the next stage of the studies, which is actually just underway from this week, will be studying older adults, and that is in two groups, the over 55s and the over 70s. And then later, when we've got a bit further through these clinical trials, we'll then study the immune responses in children.
Okay. So over the weekend, we heard about the possibility that these trials may not be able to be completed because of the dropping numbers, which of course is good news. But are there other ways we can use to identify if the vaccine is working?
Well, I think the best way of really understanding the impact of these vaccines is to test them in the field where there's natural exposure to the virus. And so one of the approaches we've taken there is our current trials, which are just getting underway, involve very large numbers of people. We will have around about 10,000 in the UK trials. And we're also now working with international investigators to expand that number around the world in the hope that in one or other setting, either in this country or elsewhere, we'll catch an upswing in the number of cases that happens naturally as lockdown is released. And that includes, as you may have heard recently in the news, that AstraZeneca, who are partnered with Oxford, are planning a trial of 30,000 people in the United States. And our intention is to try and combine the data from all these trials to try to work out how well the vaccine works as early as possible. Now, there is another approach which could be taken, which is deliberately challenging people with coronavirus. And I think this has been really important in vaccine development over the last few decades to accelerate development and also work out which of the different vaccines might actually be useful to take forward clinically. Problem with that today is that we don't have certainty about what's a safe dose of virus. We certainly have data suggesting if you give too much, you're much more likely to get severely ill or be admitted to critical care. And in the absence of a rescue therapy in that context, today is not the right moment to be doing those studies. However, I think we do have to start working towards that goal because it's certainly possible in maybe even in weeks or months from now, we will have those rescue therapies. And if is the case, and I think we can get fully informed consent from people to take part in these studies, I think at that stage, one would want to be ready to test some of these products to find out how they perform against each other.
And I think testing by deliberately challenging is a validated approach. It's been used before in vaccine development, hasn't it?
Yes, I mean, currently around the world, and that's actually mostly in the US and the UK, but some other groups in Australia and in Europe, there are very many models for all sorts of different viruses and bacteria. Here in my group, we work on typhoid bacteria, and in Oxford, there's also challenge models for malaria and influenza, and a number of others around the UK.
And perhaps finally, we're acutely aware that this is a global pandemic, and it's great that AstraZeneca is taking on manufacturing. But how accessible do you think your vaccine approach will be for providing vaccines to the population of the world if necessary?
Well, I think a really important component of the deal that the university made with AstraZeneca is sort of a guarantee of equitable access around the world. And the second is not-for-profit during the pandemic period. So that means that the vaccine development is going with the absolute intention of being able to manufacture and make provision around the world. And in that regard, work is going on with multiple manufacturing sites around the world to try to make sure that once we have data about whether the vaccine actually works, that the capacity is there in order to be able to supply. And that's a huge logistical hurdle from where we are today, where we've been aiming for that for some months now.
And sorry, just the technology that's used. So how do you grow up this recombinant virus?
So it's grown in a cell line in a manufacturing facility in bioreactors. And the challenge really for upscaling is to do this and have a process that works at the scale we use for clinical trials of hundreds or thousands of doses is very different for the scale you need for hundreds of millions or billions of doses. And there's just a technical challenge of how do you get up to that size and still have exactly the same product at the end.
Thank you very much, Andia. We're really excited to see how this vaccine develops, and well done for the work so far. Wish you the best of luck for the future with this.
Thank you very much, Julian.
Well, next I'd like to introduce you to Fran Mabesa. Fran is one of the research nurses at the St George's Vaccine Institute, and she, along with a team of others, are helping run the clinical trials at the moment. Thanks, Fran, for joining us. So, Fran, I'm very much interested in what it's like to be a volunteer on the trial. But first, can you just briefly outline what you do in the Vaccine Institute for these trials?
I'm part of the pediatric infectious disease research group, and we deal with infectious disease as well as vaccine.
So specifically for these coronavirus vaccine trials, what sort of people are we looking for to recruit?
At St. George's, currently, we are recruiting anybody who is healthy and is between the age of 18 to 55. But they should be healthy. They shouldn't have been underlying health issues such as diabetes.
So can you tell me what the process is? Let's say I'm volunteering and then I put my name, I think, online, isn't it, as a volunteer?
We've got an online advertisement, and with that online, we first give you a questionnaire to fill in. Once you get that questionnaire, if you are eligible to join the study, we'll give you a call, and if we've got any questions to clarify, that will be the time that we clarify the questions.
Okay.
Then we'll be invited to come for the screening. On the screening day, what you'd expect is we will obviously during this period we will give you a mask. We will be having the mask, aprons, and gloves. So that's how it's working these days.
Right.
And once that is done, when you come in, we'll ask you to watch a video. The reason is just to make sure that before you consent, you know what you are signing for.
So that video would tell me about the vaccine and the things that you're going to do during the trial, is it?
Yes, it will tell you about the vaccine, what you can expect once you have had the vaccine, and during the vaccine period, what you should be doing during the vaccine period.
I see.
The side effects that you are likely to experience.
And you mentioned screening. What does that mean?
We'll do physical examination. We'll collect medical history, and we will do the bloods. We want to check how healthy you are.
I see.
We want to see how your liver is doing, how your liver is functioning. We want to see your kidney function, and we also want to see if you've got a good HB, because we will be taking bloods from you. Hemoglobin,
you mean?
Yes, hemoglobin.
Yeah. Okay. Yes. So really, if you get accepted onto the trial, it's a statement that you're pretty healthy.
Yes, you're pretty healthy. Yes.
Okay. And then, how do you give the vaccine?
When you come in, we will give you the vaccine on your deltoid, which is, in simple terms, on your shoulder.
Right. Does it hurt?
It's a needle. Yes.
Okay.
So you kind of expect that most people are different. Most people, they say, I didn't even feel it go in. Some people say, I felt a bit of a sting.
And then what? How long would you keep me in for until you let me go home?
Once you have had your vaccination, you will be with us for the next 30 minutes to an hour. Just for us to observe you that you are not having any reaction with the vaccine that we have given you.
Okay.
And after that period, we will do the observations, which is pulse, BP, and temperature, which are post-vaccination observations.
So that's just to check that I don't have an immediate reaction to the vaccine. And
yes.
Yeah. And then after that, you would let me go home?
After that, we still need to set you up for electronic diary. So the purpose of the electronic diary is for you to put in anything that you can or will experience during the period, the 7 days period post-vaccination. Things that you should write in your diary is, are you experiencing any fever, any tenderness on the injection site, any itching? Are you feverish, having headaches, malaise, fatigue?
Yeah, I was going to say some of those are quite common reactions to vaccines, aren't they?
Yes, they are. Yeah. Yes, what we've got in the electronic diary is things that are common with vaccines.
Okay. After that, then you go home, and how long do you follow up for after the vaccination before the end of the study for me?
Once you have had your first vaccine, we will like to see you in the next 28 days, give or take 7 days.
Okay.
In the next 28 days, we will do the blood again to check your liver is still okay, and like the blood that we did on the first visit, liver function, renal function, and we will also do the immunology function, because now we have given you the vaccine.
Right. Okay.
And we will continue to do your blood pressure, your temperature, and your pulse. We will also collect the diaphragm that you have been feeling.
And so, is it at the end of 28 days? Is that it, or do you follow on for longer?
We still have to follow up. So the follow up will be 28 days, and 5 months from 28 days, and then from there you will be followed up in 1 year's time.
Okay. All right. Okay.
So the trial technically you will be with us for 1 year.
For 1 year. Okay. Thank you very much for that, Fran. That's really interesting and I think very helpful for people who are thinking about volunteering.
Thank you very much for having me.
Okay. So next we're going to speak to Robin Shattock from Imperial College. Robin is the Professor of Mucosal Infection and Immunity. And thank you very much for taking the time to come and speak to us, Robin.
My pleasure.
So, Robin, I understand you've developed a very new and rather innovative approach to the vaccination. Can you tell us about it and how it works?
Yes, so for this vaccine, we're using an RNA approach, and people are probably familiar with DNA that makes up your chromosomes. RNA is the message for making protein. And in some ways, we're hijacking the approach that viruses use, in that we use a sequence of RNA that can self-replicate or copy itself. And into that sequence, we insert the code for the spike glycoprotein. Now, that RNA is produced synthetically, which gives us extreme advantages in terms of producing the vaccine. And it's encapsulated into a small lipid droplet. And when that's injected into the muscle cells, it's taken up by the muscle cells. And because the RNA encodes an enzyme that allows it to copy the RNA, that when it gets into the cell, it produces multiple copies of that original message, and then starts spitting out the protein for the spike glycoprotein, which is expressed on the surface of cells. And that induces an immune response. Now, the clear advantage there is that we can use very low doses to get a very effective immune response.
Okay, so for future generations of medical and biomedical students, Robin, would you call this a live vaccine or not?
Not a live vaccine, no. A synthetic vaccine, but it has the ability to copy itself.
Okay, and that copying, is it finite or could it go on for ever?
No, it only goes on probably for a period of a few hours, maybe up to a few days. Look at the expression of protein from that construct. We see that it peaks at about 7 days and then it disappears by about 3 weeks. Okay. So it's very similar in its kinetics to a viral infection. And that might be quite important in terms of eliciting the right type of antibody responses.
Right. So this approach using RNA kind of challenges our preconceptions. So any molecular biologist from our era thinks of RNA as being rather fragile, degrades as soon as you look at it, sort of thing. But that's not the case then, is it?
You're absolutely right. I mean, RNA is degraded very, very readily, but predominantly by RNA's enzymes that chew it up. So as long as you can exclude those from the manufacturing process, it is very stable. And by encapsulating it in these lipid nanoparticles, it's then protected when it's injected from RNAs in the environment.
I see.
It's still, it still has to be stored appropriately. And at the moment, we have a frozen version. We're working on new formulations that will be stable at 2 to 8. And we can freeze dry the approach if we want to stockpile the vaccine for years.
Right. So I don't believe your trial has started yet. Can you tell us when that will be and what's your sort of research timetable?
Yes, so our plan or our timetable is to start our first in-human study on the 15th of June, hopefully, as long as there aren't any slippages, but it'll be thereabouts. And what we do is a classic dose escalation study. So we start with a dose much lower than the dose that we anticipate we'll need for safety in a single volunteer. Then we go to 5 volunteers. We start to increase the dose. And ultimately, in that first trial, we aim to enroll at least 300 volunteers so that we build up a very robust safety profile. And we also study the immune response. And the plan is that if that all goes successfully, we'd then be able to expand to a large efficacy trial of at least 6,000 people, multiple centres across the UK by October.
So that first trial is not really a classical phase 1, it's a sort of phase 1/2 trial then, is it?
Yes, absolutely.
Okay.
So it's kind of both. And doing it in as fast a possible time without cutting any corners in terms of safety.
And would you be, for that first trial, would you be restricting it to particular populations, age groups?
So we're definitely wanting people who haven't been exposed to the virus in that first study. And we're looking at 2 age groups, 18 to 45, because we know that that's where you're at your peak in terms of immune response, but also in a separate cohort of 60 to 75.
Right.
We haven't gone above 75 just because we don't want to be bringing more vulnerable people to the clinic for vaccination.
I understand. And how can people sign up for the trial? Can they do it yet?
Well, I suspect we will have a lot of volunteers. And so, some people will be disappointed that they can't immediately join in that first trial. But there will be opportunities as we scale out to a 6,000 person trial in October to have more people involved.
Okay. And when can people start to look out for the recruitment?
In early June, we'll starting to look for recruitment.
We heard recently about the problem of declining numbers of COVID patients and how that might affect vaccine trials. Do you need there to be a lot of disease around, or are there other ways you can use to detect whether your vaccine is working?
Well, of course, that's always that challenge that we want there to be less infections for the public because that's a good thing. But the fewer infections there are, the slower it will be to demonstrate that a vaccine works. We don't know, in our case, what the epidemic will look like in the UK by October. And there's always a concern that there may be a second wave of infection. We'll be very proactive at trying to prioritise recruitment in any areas of the country where we see higher rates of infection. But we are also looking at trying to set up a parallel study somewhere else in the world where incidence may be higher to increase our chances of success.
And we've also seen that there may be different susceptibilities to the disease in different populations. And I wonder if there is a possibility that there will be differences in the response to your vaccine amongst the same populations.
So we'll definitely be looking at that very carefully. At the moment, we don't see that there's any evidence that our vaccine will behave differently in different ethnic groups, for example, but we need to look at that very carefully. And I think we're very conscious that in some vulnerable groups, they already have perturbed immune responses, so they may not respond so well. And we know that the very elderly often don't make such robust responses to vaccines, so we need to study that too.
Now, Robin, your background is in HIV. You're very well known for your work in HIV vaccination development. And I know that foremost in your mind has always been the issue of making vaccines affordable and accessible to poorer regions of the world. So how does the technology you're using for the COVID vaccine address that?
Well, this is one of the reasons that we really started working on the technology anyway, is because of the issue of speed and manufacturability. So we already are able to make the equivalent of 2 million doses in a litre of material.
Wow.
That's, traditional vaccines would need 1,000 litres, maybe 10,000 litres, a large factory. So we anticipate that as people get more familiar with the technology and with the manufacturing, you could have very small manufacturing plants in different parts of the world. And that we think would help with vaccine equity. It would also help with future pandemics because different parts of the world could start manufacturing the same vaccines simultaneously.
I imagine because it's a synthetic vaccine, it's actually quite easy and quick to make and scale up.
Yes, it is pretty easy because you can have a relatively small footprint. You could do this in the equivalent of 2 shipping containers. You still need the training, the expertise, the ability to show that it's a vaccine of high quality for human use. So those things don't change, but the infrastructure that's required is much easier and of lower cost.
Well, it's fantastic to hear about this new technology and how quickly it's been taken up in the fight against COVID. So, Robin, good luck with your work and really hope you do well in the old clinical trials. Thanks for joining us.
Thank you, Julian.
I hope that's provided you with an interesting insight into how vaccines are made and the progress that we're making in COVID-19. What I want to impress upon you is that the advances that are being made with these 2 vaccines in the UK has really been achieved in record time. This is a process that can normally take 10 to 15 years and has been condensed into a period of just under a year, whilst trying to maintain the quality and the safety of the vaccines that we all expect. What you haven't seen is the huge number of people who are working so hard behind the scenes, the scientists, the doctors, the nurses, and of course the fantastic people who volunteer for the vaccine trials. And we shouldn't forget too the people in the pharmaceutical industry, without whose experience and expertise none of these vaccines would ever get made. If you'd like to find out more details about the Oxford vaccine and the Imperial vaccine, and also the trials going on at St George's and elsewhere, then these are the websites that you can go to. You'll also find details on those sites about how to volunteer for any one of the clinical trials that are currently recruiting. St George's scientists are carrying out research in many aspects of coronavirus, not just the vaccine. And if you'd like to find out more about what we're doing, then you can find the details on this webpage. And if you're able to help, then this webpage will take you to a site where you can make a donation to our Coronavirus Research Fund. Finally, I'd like to thank all the people who helped put this video together. Do keep an eye out for our other Spotlight on Science events, and you can find out more about those at this web address. Thank you, take care and stay safe.